Welcome students. Welcome to the first brand new video series of Biotathya Academy. In this video lecture, I will be giving you a detailed knowledge about the chapter Introduction to Microbial Diversity, which will include Whittaker's Five Kingdom classification and Carl Woese system. This chapter is included into the university syllabus of Calcutta University, that is the first semester syllabus. In this video or in this video series, you can say, there will be numerous playlists, and each playlist will deal with the particular semesters, and each semester will have different chapter wise detailed notes and detailed knowledge about each chapter. So starting off with the first chapter, this will be on Calcutta University, but this channel will not only give you knowledge about Calcutta University, but other universities as well. The syllabus will be similar to almost all the universities will have similar kinds of syllabus structure, but some chapters may differ from each syllabus to syllabus. But as of now, I am going to start with Calcutta University semester 1, that is Introduction to Microbial Diversity. I am Priyanka Shah, and I have done my MSc and BSc in microbiology, and I have also been a researcher in VIT Vellore from the past 3.5 years, doing my PhD in neurobiology. So I will be your educator and mentor. So let's start the journey. Please be with me. If you like the videos, do subscribe to the channel, and also do like, comment and share, and let me know what kind of videos you like most. So without further ado, let's get started. So coming into the topic, first we have to understand what is microbial diversity. What does the term microbial diversity means actually? Microbial diversity, from the term we understand, is the diversity of microbes. It is the diversity of microbes. Now if I talk about microbes, microbes doesn't only deal with bacteria, it deals with different kinds of microbes. It can be protists, it can be archaea, it can be fungi, it can be any other unicellular creatures, that is viruses as well. Most probably, when we talk about microbes, we basically understand it as bacteria, but this is completely a wrong concept. This is completely a wrong concept. Microbes doesn't only means bacteria, it means it consists of protists, it consists of archaea, fungi and other unicellular creatures as well. So when we are studying the diversification of these microbes, we have to study what kind of diversification bacteria has in this earth, protists, archaea and etc. So the biosphere is populated by a wide variety of bacteria. If I omit the term bacteria and write it here microbes, that will suit much more. That defines the parameters of life and establish the conditions required for the evolution and survival of other organisms. Now here comes the major point, or the main point. When we are talking about the diversification of microbes, it also helps in the evolution and survival of other organisms. For example, if we talk about humans. Now, you will say me that, ma'am, why does the evolution of humans, or why does the survival of humans related to the diversification of microbes? Now, let me tell you, students, in human body, we have the gut microbiome. Just this is an example, general example I'm giving you. We have the gut microbiome. In our gut microbiome, there are thousands and several microorganisms residing. Okay, the specifically bacteria. So these bacteria, these survival of these bacteria, or these inhabitants of these bacteria is very important for the survival of our gut, or the healthy, or if we talk about a healthy gut, that means that the gut is composed of healthy amount of microorganisms. So obviously we can understand that this microbial diversity is hugely related to the survival of organisms like humans. Now, various types of microorganisms, again, point to be noted, not only bacteria, protists, archaea, fungi, or any other unicellular creatures, can be divided into groups. according to the diverse genomic structure. Now here comes a few basic characteristics upon which you can divide the microorganisms, such as, first if I give, that is the genomic structure, what does the genome contains, then the evolution, then expression like the protein expression, then their diverse physiology, that is the fourth one. The fifth one is the cellular metabolism, and the sixth one is the morphological characteristics or the morphology of the microorganisms. The multilateral idea of microbial diversity includes microbial genetic diversity, microbial species diversity, and microbial ecosystem diversity. Now, in the upcoming slides, I will be teaching you about the different microbial diversity, which is divided into the on the basis of multi-latitude or the area specific. That is genetic diversity, species diversity, and ecosystem diversity. So, on these particular factors, you can classify microorganisms. On the basis of these kinds of diversity, on the basis of these three kinds are the broad categories, you can say. So, these broad kinds of categories, you can divide microorganisms. Now, coming into the concept, already I have discussed. Still, here is a basic definition where we can say that variety of microorganisms found in different environments, differing in form, function, genetics, and ecology. Major groups include bacteria, archaea, fungi, protists, and viruses. Okay. Now, bacteria are prokaryotic and they are ubiquitous. Ubiquitous means that they are spreaded all over the earth. Archaea means they are also prokaryotic in nature, but they are often extremophiles, that they can grow in extreme conditions. Then fungi, these are basically yeasts, molds, decomposers. Then protists, that are unicellular eukaryotes. You have to keep in mind they are unicellular, not multicellular. And viruses are acellular infectious particles. Okay, they are acellular when they are outside the living host, and they become live or cellular infectious particle when they enter into the host body. Now, the concept of microbial diversity can also be divided onto morphological, physiological, genetic, ecological. And the significance is that microbes drive different kinds of nutrient cycle, maintain different kinds of ecosystem. It can impact our health. It can drive the power biotechnology. Now, coming into the three classic levels of biodiversity, the three classic levels of biodiversity are genetic diversity, species diversity, and ecological diversity. Genetic diversity. From the term genetic diversity, what can we predict, or what can we guess, or what can we understand? As the term genetic comes, we can understand that it is somehow related to our genes. So, obviously, the diversification of genes in our body will be genetic diversity. So, let's see what this definition says. The definition is saying that genetic diversity refers to the variation in genes, as I said you, within a single microbial species. Within a single microbial species is like, suppose B subtilis. You all might be knowing that B subtilis, Bacillus subtilis. So subtilis is a species, and if we talk about this particular subtilis species, in this particular species there can be variations of genes, and then from those variations arises strains. What? Strains. So if a species kind of varies from one gene set to another, then it lands up into making different kinds of strains of species. It's what allows microbes to adapt and evolve rapidly. It can evolve rapidly. For example, here they have given an example that bacterial species can have many different strains, as I have already told you, each with unique genetic traits. They have particular genetic characteristic features. Now, if you tell that, ma'am, I'm confused between the term traits. What is traits? I'm just giving you a basic example. Traits are basic characters. Like if I tell you eye color, that is a trait. And if I tell you blue or black or brown, that is the allele. So if you misunderstand between traits and alleles, then you can easily get into the point that traits are characters and alleles are the color. For example, the eye color, if I tell about. So eye color is the character and the color of the eye is the allele. Enable it to survive in different conditions or resist antibiotics. So as I was telling you, if the gene set differs, then the gene will differ and the particular strain will also differ. So this particular strain, suppose one strain can survive in one condition but not in the other. So different strains can survive in different kinds of conditions or resist antibiotics. What happens if this genetic diversity is like what happens if this genetic diversity basically is possible in bacteria? This helps in the adaptability, and this is a key reason that microbes can thrive in virtually every environment. In every environment we can see a set of microbes. From boiling thermal vents to frozen polar regions, we can see microbes. Genetic diversity means microbes of the same species. For example, E. coli. Coli is the species, may look similar but have different genetic codes, which gives them different abilities. Like it can be harmless or it can be harmful. Let's go into some examples, then it will be much more clear. For example, if we talk about Escherichia coli, it has different strains. For example, non-pathogenic strain, if we talk about, it is K12, that is harmless and used in labs. Then if we talk about the pathogenic strain, it is O157 to H7, which carries an extra gene for toxin production, that is the Shiga toxin. This is a hugely impacted and a highly risked toxin that E. coli produces. Particularly, this strain can only produce this toxin. Other strains such as K12 or the other strains cannot produce this toxin, and this toxin can cause food poisoning. Same species but genetic variation gives very different strains. Now, if we talk about Mycobacterium tuberculosis complex, this can differ in different strains. Some can be drug sensitive, some can be drug resistant. And if we talk about viruses like RNA viruses like influenza or SARS-CoV, the chances of mutation rates are very high, and for this reason the genetic diversity rate is also very high for the case of viruses, because viruses can mutate itself, and for that reason making vaccines or producing medicines is very high and is very difficult for viruses, because they show variants and they can alter their transmissibility or they can escape the immune system of our human body. Now, if we come into the second one, that is the species diversity, what does the species diversity mean? Species diversity is nothing but from one species to other when there is diversification taking place. That is basically species diversity. It is very much straightforward. There is nothing any complex in complexity that is in between this. It refers to the number of different microbial species in a particular community. Like for example, Escherichia coli, or if you talk about B subtilis, so this subtilis can differ. This species can differ. Variety of microbes on earth is staggering. Scientists estimates there could be an over trillion microbial species, most of which are yet to be discovered. And this incredible species richness is what makes microbial communities so robust and resilient. I hope you are understanding. Now, if we come into the examples of species diversity, we can see that if we talk about the genus Bacillus, we can see that it can be subtilis, which is a non-pathogenic model organism. Now, what is model organism if you say? Basically, in labs, when you will perform any kinds of experiments or any kinds of experiments are performed with this particular genus, then this particular species is used because this is non-pathogenic and harmless. Then there can be anthracis, which can cause anthrax disease, Sirius, which can cause food poisoning, and it is also a soil bacterium. Then if we talk about the genus Streptococcus, it can be pyogens which cause strep throat or strep throat infection, then there can be scarlet fever. Then if we talk about pneumonia, it can cause pneumonia, meningitis. Then if we talk about mutants, it can cause dental infection or dental caries. If we talk about the genus Aspergillus niger, it is used in the citric acid production. If we talk about flavors, it can produce aflatoxin or toxin. If we talk about fumigators, it can cause opportunistic. It is an opportunistic pathogen, that is, when it will get the opportunity, it can cause the harm. If we talk about Vibrio, there can be cholerae, which can cause cholera. Parahemolyticus, which is a seafood, which can cause seafood poisoning. Vulnificus, it can cause different kinds of wound infection and septicemia. Coming into the last but not the least diversity, there is the ecosystem diversity. Now, ecosystem diversity is basically the diversification of not only microbes plus with their environments, microbes plus the environments. So along with the environments, when the microbes evolve, that is ecosystem diversity, where they inhabit. Microbes form complex interdependent communities. Microbes can form complex interdependent communities which are called as microbiomes. Now, you don't have to worry about that. I will be dedicating a particular slide on this microbiome, an example of microbiome in my upcoming classes and upcoming slides. So keep and watch on this channel, and please hit the subscribe button so you can get the detailed updates on those, so that are crucial for functioning of larger ecosystem. For example, here they have given an example of ecosystem diversity. There is the human gut microbiome, where not only the microbes are responsible, the gut is also responsible for the diversification. It is a diverse community of microbes that aids in the digestion process and protects against pathogens. Similarly, microbial communities in the soil are essential for nutrient cycling or plant growth. This deals with the soil microbiome. So, the specific mix of microbes in a given ecosystem dictates its function and stability. What kind of functionality or stability the ecosystem diversity or the ecosystem will have, it deals with the kinds of microbes and kinds of ecosystem it deals with. Now, going into the example, if we talk about extreme environments where the temperature conditions or where the other factors are extreme, for example, Thermus aquaticus, where um, Thermus aquaticus is an enzyme which can inhabit in hot springs where the temperature is highly, very much high. Those are thermophiles, which are also a source of Taq polymerase. This is an enzyme used in PCR. Then Halobacterium salinarum, this is basically found in salt lakes. And Acidothiobacillus ferooxidans, which are found in acid mine drainage, and these are acidophile because these are acidic. They can tolerate acidic pH. Okay. Then soil ecosystem, if I talk about, you might be hearing about this particular bacteria, Rhizobium leguminosarum, which stays or which resides in the root nodules of leguminous plants such as pea plant, and they can help in the nitrogen fixation. Then Streptomyces griseus, which can produce antibiotics. Bacillus subtilis, which is identified as a decomposer and a spore former, because when the conditions are not favorable, they can form spores and they can inhabit in that. Then if we talk about aquatic ecosystem, there is Prolococcus marinus, which is a photosynthetic cyanobacterium. Then Vibrio alligoniticus, which is a marine bacterium. Then diatoms like Thalasseria pseudomonas, which are the primary producers. And if we talk about the human body ecosystem, like the microbiome, for example, the gut microbiome, the most important bacteria residing is the Lactobacillus acidophilus, then Staphylococcus epidermis, which cause epidermatitis in skin, then Streptococcus mutans, which causes oral cavity or oral infection. So, in the next slide, we will be dealing about what is microbiome. That will be continued in my next class. So, for this class, I have taught you about the three kinds of ecosystem and what is the diversification of microbes means, and have dealt with different and various kinds of examples so that you get a clear cut idea about that. You can also visit my website biotathya.icu, where you can get detailed information about the academy. And for admissions, as I take online classes, for admissions you can call into this number or WhatsApp me into this number, and you can get information about that as well. So for now, I'm leaving here and I'll continue my next topic in the next class. Till then, take care and bye. And let me know in the comment section what you need next so I can make a dedicated video on that topic as well. Thank you.